Hi, everyone. Welcome back to the session. I just want to know how many of you were able to set up the collab and have a good Wi-Fi connection. Okay, cool. Um, for those who was able to set up collab, please follow the collab on your end. I do have some issues with Wi-Fi connection, so I was keeping so it just kept disconnecting. So for my end, I would just run through a local notebook, but the code are the same. So normally the results should be also the same. And also for this session, the plan is to go through the notebooks all together. If you have questions, either you can post question on the portal page for the lab one, or is there anything really blocking you for understanding? You can also stop me anytime just for clarification. Okay, I see everyone got a seat, so let's get started. Uh, so for this coding session, what we would aim for is that we take a EGFR as example, what Cas has introduced earlier on. So our goal in this session is we want to understand our training data, the data set. We want to we'll demonstrate the molecular presentation in practice and hands-on, and then apply machine learning algorithm to train models, apply this model to do virtual screening, and then followed by molecule prioritization. So here, the example here is EGFR. And for the sake of this tutorial and also for a better understanding for broader audience, we choose, so this session, we set a very rather easy objective for virtual screening. So basically what we aim for is to find to leverage public data set and then train model. The ultimate goal is to find the novel inhibitors for EGFR within the scene within the scene chemical space, and then we want to prioritize prioritize about 100 molecule with diverse with certain diversity and within a molecular weight between 218 and to 400,000 for further study, which will follow by the lab from tomorrow on. So, okay, let's start with the data. So as Cas presented earlier, so if you don't have the luck, have your own private lab to generate your own data. So what we can do is we can try our best to leverage data from public domain, like from Campbell, from PubChem, or other database like Binding, or other more specific database like for kinases or for gene expression data. So in this lab, we focus on EGFR. The easiest thing to do is that we can just simply type EGFR inhibitor in Campbell, and then it returns you a long list of molecules with different readout and various type of assays. And what we often see as a readout for the EGFR inhibition is, for example, the percentage of inhibition, and also followed by at different concentration. And then another typical readout is IC50, and this is actually it's literally the concentration of a molecule which exhibit 50% at its maximum inhibition capacity. And this is calculated based on the curve of IC50, based on the what I just mentioned, the percentage of inhibition at different concentration. And naturally, so we want for a drug, the lower the IC50, the higher the potency, the more potent the inhibitor is. And so this is a minimization problem. And but in practice, a lot of people use PIC50, which is a negative of logarithm transformation of IC50. The reason why people rather choose this is that because it scales the IC50 in the distribution which I show here on the left. And it's much easier for the machine learning algorithm to learn from, and also it's more intuitive for people to understand about the inhibition ability of a compound in comparison to IC50, which might cross a really big range from very low value to very high value. And as you can see, the IC50 distribution is much more skewed to the one side. Okay, there's another type of readout, which is KI/KD, which literally people measure the kinetics of kinetic inhibition of the compound. But this is more used for the programming later in more advanced stage, because this kind of readouts are often more accurate compared to IC50. So that's a different type of a readout. And now for this session, we have already prepared a toy data set we can play with. So here in this file, if we load, are you able to load the file and go through to play with the code? Okay, so if we load this file, the most important column from this file is the molecular column for SMILES. SMILES is, as you know already from during the day, so it's a string presentation of molecule. And then another one is the inhibition column for PIC50. And we will focus on modeling on PIC50 today because of the advantage I just talked about earlier. So here in our data set, we have about more than 4,000 data points, which is already quite a lot for a drug discovery program. So something I want to mention here. So here is just a toy data set, but in practice, once you have so many data from, for example, PubChem, we have to be careful to choose which bioassay for your problem, because we cannot blindly combine those bioassays. You should always choose the ones which are most related to your problem and which are most comparable with each other. So here is just a toy data set. So we just let's continue from here. So for PIC50, so normally people use a PIC50 cutoff value to identify inhibitor and non-inhibitors. The range could be from 5 to 7, sometimes even higher depending on program. And here in this tutorial, we use 6 as a cutoff value. That means everything greater than 6, we identify as inhibitors. Anything below or we're defined as non-inhibitors. And the PIC50 equals 6, which means the IC50 equals 1 micromolar. So now let's look at the data distribution. So as you can see, because for this toy data set, we have kind of rather balanced distribution, although maybe on the left side we have a bit more compound molecules, but it's definitely enough for us for modeling. But in reality, in real drug discovery problem, the distribution might be skewed more on the left, sometimes on the right, depending on the stage of the program. And now we know the distribution of the measured value. Now let's take a look at the structure and how the molecule are distributed within the chemical space. So early on, we have already chosen the library we would like to screen against. That means, so now what we are doing is that we visualize the chemical distribution between our data. So basically the training set, I would call it, and then also the library will screen against later on. So the data is highlighted in green, the libraries are in orange. So and now we can see actually the library, the chemicals distribution of the library covers pretty much our data set. And so this distribution was computed it's a UMAP based on the molecular feature and within a reduced dimension. So what we can learn from here is that just want to remind you that because our goal, again, we made this sample, so we want to know we want to discover novel inhibitor within the chemical space of our training set. So the green part. That means, so whatever we train from based on our data, a big portion of the library, the molecule in the library, would fall into the domain of the applicability of the model we will train on. Okay, so that's some basic data analysis and statistics of our training set. So here, I just so here is, we talked about during the day about the molecular representation. Here, I don't want to repeat all the theory that people have already introduced. I just want to show you hands-on how it would look like in practice when we code in Python. So here, for example, we first start with Morgan fingerprint, EC50, ECFP. So this Morgan fingerprint was already implemented in RDKit, which is a Python is a library designed for chemoinformatics. So this implementation is also available in Morphit. So for those who doesn't know about Morphit is an open source library developed by Valence, which is also a hub for all types of molecular featurizer that you can find. So basically, you can just simply import the fingerprint vector transformer and then specify the type of fingerprint of your interest. Here in our case, it's EC50 with a radius of 4 and with a fixed length of 2048. And then what you need to do is you take your smiles from the data, then just call the transformer and compute the features, the fingerprint. Okay, let's give it a minute. Yeah, so here this is what the feature would look like. The fingerprint would look like for each of the molecule in the data set for about 4,000 molecules. And as we know that because the fingerprint normally is very sparse, so it's expected that we see a lot of zeros. And then we can also get inhibition values from the data set. And so here, once we get both the fingerprint of compounds and the label, which is also the value of the PIC50, we can take it as an input for our machine learning model and and start building models. Okay, that's the example for fingerprint. Now, let's move on for molecular graph representation. So basically, what we need to do once we get a molecule, for example, here is in string format, we need to convert the smiles into a RDKit molecular object. And you can do that by just pass through data mole, which is another specialized library to deal with molecules. And then the second step is that we would like to, in this exercise, we would like to handle all the graph in PyTorch and geometric graph object. So the second step is to use PyTorch PyG graph transformer, which is also available from Morphet, and convert the molecule to graph object. And now let's take a look at an example of one graph object in PyG data object. So basically here, the X represents the feature of the nodes. So the graph has feature on the nodes, feature on the edges, and then the edges which represent the connectivity of the graph. We have already heard during the course earlier. So here in this, so in this data structure, it contains the feature for the nodes and feature for the edges, and then the information about the edges and which you can visualize as adjacent matrix. So here, that's the example for molecular graph, and we can take this PyG graph data object as the input to build the GNN model later on. And so now we have our data, we have our label, we have a good appropriate molecular representation. So now we can move on the modeling part. So before diving, start training the model, let's step back and first decide and make a decision on the evaluation strategy, which include the metrics we're going to use and also the train test split we will use. Also, as earlier mentioned by Cas, that the decision of the split really depends on your program, the goal of your program, and also your data, and also the deployment set, which means here the screening library. And today for this tutorial, we would train regressors for the inhibition prediction. And we would like to predict the continuous value and use that value to rank the molecules. So the typical typical metrics for regressors are what as you can see here, Pearson R, Spearman coefficient, mean square error, and then because often people also use regressor to deal with classification tasks, because once you have a well-defined cutoff value, so here we also track the metrics like precision, recall, and F-score. So what we're going to do at first is that we let the model train on the single training set, and then evaluate the model performance on the test set. Okay, that's the metrics part, and now let's talk about data splitting. And again, I have to emphasize, so for this exercise, we do hit identification. And we want to discover any novel inhibitor within the known chemical space. And for that purpose, one of the most used splitting, so either we can use random split but here we will use another well-known splitting method, which is called scaffold splitting. So again, scaffold of the molecule is the core structure of a molecule. So the idea of scaffold splitting is that we group molecules based on their scaffold, and then we we let the model to train on subset of scaffold, and then we hope that, and then we make the prediction on another set of scaffold that the model hasn't seen. And our hope is that the model can generalize well on the unseen scaffold and chemical region. And for doing that in practice, I encourage you to, as this example, we have Splitto, which is also another open source Python library developed by Valence, so which offer different types of splitting algorithm. And here you can just simply call import that library and get the correct splitter. So again, for the scaffold splitting, one tricky part is that you have to make sure that there are enough different scaffolds in your data set if you want to do generalization. Because, for example, if you have only one type of scaffold, because your program is in the very late stage, you have only one molecule compound series, then it's going to be hard if you want to generalize on new unseen scaffolds. Also, like even sometimes 2 or 3 wouldn't be enough. So let's take a look at our data set by getting the number of scaffold. So we have about 1,700 scaffolds for more than 4,000 molecules. So we would say we definitely have enough scaffold that we can train on. And now what we do here is that we call the scaffold split from Splitto, and then we define the test set. Maybe sometimes we can set the random state, and then we get one train test split just for demonstration purpose. So here we have 3,000 molecule in the training set, another 1,000 in the test set. We're gonna train on the training set and evaluate the performance on the test set. But before training, let's look at the molecule distribution in the chemical space between training set and test set. Again, here, green shows the training set, orange shows represent the test set. As you can see, they are pretty, they're kind of spread all over the place between training set and test set, which indicate that the scaffolds in our dataset are very diverse. Although you can still see some clusters here and there, but still, in general, it's very spread out. Again, maybe you see some dots between training and test that they are very close. Again, this is a reduced dimension visualization. So in reality, maybe they are far away if you import them multiple components and multiple dimensions. Okay, here we have already talked about we have already our training set and test set. Now let's start training a random forest, which is always a strong baseline in machine learning methods. Okay, let's, so here we just, let's, okay, sorry, I forgot to run one cell. Sorry, I have to get back. Yeah. Let's get both the feature and the label of the training set and test set. And then here we define the random forest regressor with some basic parameters and start. Then we do the fitting, which won't take long. Okay. And now the model was trained, then let's look at the performance on the test set. Oh, oops. Oh, sorry again, this was not around. Okay, so here we see the value of the metrics across for both regression tasks and the classification tasks. They look pretty decent. And then also we see the correlation between the prediction and the true label, the value of inhibition, the PIC50 value. They are so far looks the model looks okay. And something I want to bring attention here that we see the mean square error is 1. It looks pretty low, but then we have to keep in mind we are training on PIC50. And do you know, like, if we have made a decision, like we have the difference between PIC56 and PIC5, it's a huge difference. That's why when we apply this model, we definitely have to pay attention on the value we are predicting, because yeah, like a PIC50 of 5 would represent. If I go back, PIC50 of 5 is here, and if we're aiming for PIC56, here there's a huge difference, and we might include a lot of negative false positive values here. Just keep that in mind. So sometimes when you see a really nice regressor with beautiful metrics, doesn't necessarily mean that you can just apply the blindness and select molecule blindly. Okay, so we have already trained the model on one train test split, and the metric looks good, but is that by luck, or because the model just liked the test set we selected? So to avoid that kind of bias, and also to have a better access of the model performance, and as you all know, as machine learning scientists, we often do, we would like to access the model performance across different types of split. And in this case, we use scaffold-based splits again. So here's a piece of code just to run the cross-validation process. So basically, we train on one split and test on the test split, and then loop over the entire splitting list. Okay. So for this, this might take long. So before running this, I will just explain the result, because this cross-validation process will take some time. Later we just wait, and then we can continue to explain the next sessions. So here we have already run the cross-validation. We compute the mean value of each type of metrics, and we also check the standard deviation, just to check how the model perform differently across different split. And here's the box plot. Besides of MSE, the rest metrics are pretty, the value are pretty close, and so we can kind of draw a conclusion here that model looks perform pretty stable in a stable way across different split. And maybe we can use this model for virtual screening for our purpose in this virtual screening scenario. Okay, that was random forest, the baseline. And now, so for this tutorial, we also want, during the day, we have also talked to GNN. So now let's try build a GNN model with the graph, molecular graph representation we just talked about. So basically, the idea is we have a graph object, molecular graph object as input. We build here, which we would like to train on train a graph neural network with the 3 convolutional layer, and then followed by a linear transformer for the final prediction on the PIC50 value. So here is where we define the GN model. So here, and I have put a piece of code to define the GN model. So basically, this GN model is based on the implementation from PyTorch geometry. So PyTorch geometry is a library that it includes many different types of GN model architectures, which is really convenient to use. So for this, if you want to look more details on the GN model, you can later on you can just open this file and have a look. I'll also go back to the page of the PyTorch geometric page, but probably you guys have already know about this library and know how to use them. Okay, so here we have the GN model defined, and now we define our optimizer for parameter updating, and also, of course, the loss function. Okay, here we have our. Here we've utilized the defined GN model, and then again we transform our dataset into graph object that which can be used directly as input for GN model. Just something to pay attention that we have to again to assign the label value to the graph. Again, as any deep learning models, so we often deal with the data with a data loader for the convenience, especially if you want to tune on the patch size and define the other parameters like whether shuffle the data or not. So here we do the same. Here we are doing the same thing. Okay, here below is a sample training loop. So basically, we just iterate each batch of the training set with the data loader, and then yeah, let's iterate the data 5 times. So by defining the number of epochs, and now let's start training sample training. Okay. The model has finished training. Now let's look at the model performance. Okay. See again, GN model failed terribly compared to the random forest baseline. I mean, kind of we're not surprised because now we're using just one type of parameter and we didn't do any hyperparameter tuning, right? And also in general, we know that neural networks perform much better on the large data set. And here we have only kind of a small size of 4,000 molecule. So again, here is just for demonstration. We just want to show you guys how to use our and any public available PyTorch code for modeling the inhibition. So in practice, we would perform hyperparameter tuning. For example, batch size, on the learning rate, weight decay, and of course sometimes and other relevant parameters, including so for those who are not familiar with hyperparameter tuning, so it's basically the idea is to search over a big space of parameter sets, and then try to find the best parameter setting for your model, for your data, and for your objective of the program. So there are multiple ways to do that. You can do research. You can also do some more advanced, clever way, for example, Bayesian optimization. And there are very very well developed framework which allows you to do that, like Optuna, and also the WenDB sweep, for example. But in this case, instead of only do train test, normally we would also have additional data set called the validation data set, which allows you to do the hyperparameter tuning and for model selection. Okay, so now we have models. We have trained a random forest regressor. We have also another graph-based GNN model, which predict the PS50. And of course, in this scenario, we'll pick the better model, at least which looks like the better model, which is the random forest regressor for our virtual screening. So here we will load the virtual screening library. So I have already loaded earlier on. So you can, so it's a file with SDF5. It's a SDF file which includes all the structure and 3D coordinates of molecules. So you can just, instead of using RTK, you can also choose the data mode, which loads SDF directly just by a single line code, and assign the molecule column directly. So here we have 24,000 molecules in this library, and in this library we have more smells, which is smells representation of the molecule. We have also already integrated molecule object, and then the catalog ID, and another basic molecular properties like molecular weight, C log P, and TPSA for extra. And normally this kind of property we can use later on for filtering the molecule, depending our interest and the need of the program. And as you can see, one of the filtering we would apply here is the molecular weight. So in this library, we have a molecule with a small size like 177 Dalton or until 544 Dalton. So that's what we're gonna pay attention later on. So we have loaded our library. We have all the molecules. The very straightforward thing to do right now is to we use our selected predictive model and then make prediction on those unlabeled molecule from the library. And here we define the scoring function. So basically, what it does is to first get compute the molecular feature, which will be fed to the machine predictive model, and then what we do is to simply predict the inhibition value of the molecule use our predictive model. Now, once we have so here we have a rather small library, but in practice, oftentimes that we would deal with a much larger library to increase the success rate of finding the new hit. So normally, and in that case, that we would like to parallelize the scoring procedure in which also allows to fit into the RAM and for other computational limits. Okay, now we have our library with the predicted PIC 50 value. Okay, now what we will do next is, of course, we have all these molecules scored. Now we want to prioritize the most promising molecule, which we would like to test later on in the experiments. So let's have a look at the distribution of the predicted PS50 value. So as you can see, everything from the on the right side of the yellow bar can be seen as predict as inhibitor if we use a threshold of 6. And then we can see very few number of molecules which if we use a higher threshold like 7. So again, I just want to remind that so earlier on we said we will use 6 as a threshold in this for identify inhibitor and non-inhibitor, but just do you recall that earlier on we said the MSE value, so the mean square error of the regression, the random forest regression model is 1. The average value is 1. So if we really use the 6 as the cutoff value, there's a highly chance that we might get a lot of false positive because of that average square error that the model might be wrong at. And this is very important in drug discovery program because these errors are very expensive. In that case, we would like to increase our threshold a bit more. So we use 6.5 as a threshold here, just to de-risk the failure for our drug discovery program. And then another criteria we have said early on for this exercise is that we want to limit the molecular weight. So here simply because the molecular weight is already computed. So basically we just apply a simple filter on the molecular weight, which resulted 145 molecules in the candidate pool. Again, something minor, sometimes people might forget about is that you might have the same molecule in the screening library in your training set, because most of the case when people start program, they will firstly screen, make a hit identification program experiment on a medium size of screening library, and then they leverage from there. They start the program from there. So just in case, just trying to avoid repeatedly testing the same molecule, we need to remove those molecules from the prioritization set. So here, one easy way to do is that just by computing the unique ID. Here, we can just simply use the DataMore API DataMore unique ID, which is actually a hashed ID based on the intricate key. And here, obviously, we have spotted one molecule from the training set. Okay, so now just let's look at the distribution of PS50 in our prioritized set. So we have a few molecules have a rather have a PSV greater than 7, and in total we have like 144 molecules. And let's visualize them in the chemical space. Okay. So the green dots represent the training set, and the orange are the molecule that we have already prioritized so far. As you can see that we have some, if you can see clearly, we have some molecules spread apart from each other, but as a majority of the selected molecules are actually in the middle, in they are rather close in kind of in one or two cluster. So as what the Cas have mentioned earlier on, like we to decrease the failure rate. So we don't want to repeatedly test molecule depending on the program again. So for the early on stage and for the purpose of this exercise is heat discovery, we don't want to repeatedly test molecule sets which share the same scaffold over and over again. Because in the worst case, this scaffold would definitely fail, and then we are wasting our energy and the resource to those molecules which are not really useful in the end. So for that purpose, we would like to just pick a diverse set of molecules from this candidate pool. So first of all, we would like to do that. First of all, we would like to rank the molecule by the predicted PIC50 value. The reason is that, of course, when we pick the most promising one, we always want to keep the most potent molecule. So in that case, so for the diverse pick, we can use directly datamol pick diverse function, and then we set number of molecule. We want to pick 100 molecule, and then we definitely keep the first at the top most potent 5 molecule from the set. And then we just run that. Okay, now we have selected 100 molecules. We want to display them again in the chemical space just for verification. Okay, compared to the chemical the plot earlier, so here the molecules we picked are much more spread apart instead of falling into one single cluster. So that's what we are aiming for. Okay, so then here we have our final set of 100 molecules that we want to test later on. Just let's look at those molecules in practice and just see how they look like. So normally once we're at this stage, if you're lucky, you have a medchem expert in your team, normally it's their job to tell you which molecules are good or which are definitely a no-go. That, and maybe very likely, a lot of time the medchem will tell you, oh, your molecule sucks. And then it's going to be embarrassing. So for our end, what we want to do then in the next step is that we want to make sure that the molecule we prioritize have a higher chance to go through the selection funnel and move forward for the later on stage of our program. So in that case, we would like to select molecules which are more drug-like or lead-like. And in that case, we would encourage you to, normally we would like to apply a series of medchem filters, for example, to prioritize those molecules again, at least remove the molecule which definitely has red flags. For example, they have some toxicity flag in the past, or some substructure is definitely undesired for your program. And for that, we have another open source library called the medchem, then that we often use in our own at valence for our own program. So I would encourage you to check it out. But of course, still, you cannot apply those filters blindly. It's always good to consult a person with chemistry expertise and help you select those molecules. Okay, that's the virtual screening session, and so now we have already have picked our 100 molecules, and so for tomorrow's lab, so my colleague will show you how to do the structure-based screening by screening the same screening library. And it's going to be interesting if we can compare what they have selected and what we have selected here. So thank you for your attention. And if you have any questions, please shoot it now. Thank you.
Any questions? I'll bring them at you.
Okay. Yeah. Do you want to come?
When you made the diagrams visualizing the chemical space, what do the axes actually represent?
Okay, so here, so basically this is like it's a UMap with it's a UMap that basically we use the molecular feature. So for example, like a ECFP or other features of choice, and then we let the UMap, so UMap learn is a library to do that, and basically it learns the data structure in a lower dimension. Then, for example, here is like a one dimension of the learned feature, and this is another dimension. So, but in general, it's more like a high dimensional problem. But in 2D, we just visualize 2. And this is one way that how we visualize the structure of our data.
Any other question?
Yeah, so it feels like the hard task here, which we didn't I feel like we kind of cut short is like the reason why this works so well is because we have data which is very shootable. And so like I think it was 24,000 or whatever. How do we go about selecting data which would yield those results? Because I feel like that's like selecting that subset of training data is really what makes this whole thing work, right? And it's like the paramount step and the first step. And so could you expand on how we would go about choosing suitable data?
So you're talking about choosing suitable data in general from public domain, or?
Yeah, so like the data set we've used here is like that screening library. How we the what? Yeah. So how we go about choosing that subset of that library, like that data?
How did we choose the library based on our data? Okay, first of all, we choose this library. One of the reasons is for the sake of this tutorial. We want to reduce the size for screening. And also another reason is that, again, the whole context of this exercise is that we want to find the ATP binder. That's why we narrow down instead of having billion of molecules, we narrow down to the library which is specified for ATP binder. And also another reason is that this is a rather easy, one of the easiest scenario in drug discovery. It's not like EG-verify is easy. It's just the goal here. We try to set is easy because what we are aiming for is just to find the inhibitor within the chemical set of the training set. But in practice, even though here the molecule, the library set here covers pretty well the the training set, but you can still see a lot of the molecule which are not really falling into the space of training set. That means if depending on the goal of the program. For example, if your goal here is you want to completely move away from the training set, you want to have a model generalize really well outside of your training set, the training of the training set. For example, we have a trend here. I want to have a model really works well here. Then it's another scenario, it's another story. Then we have to take into account the other distribution problem. Then it's another scenario, how we select the data splitter, how we evaluate the model. And regarding how we exactly choose the screening library, again, depending on your goal. And for example, in your program, you want to for example, you want to move away from pattern space. So, and for your company, you have already available screening that which we have already compound there. You can test on. You want just screening on that library. Then, of course, you should also compare your training set and to that library, and then select the correct model validation strategy. Did I answer the question or not? Sometimes the library is not. If we have a complete freedom, we know what we are looking for. We just choose that library which fits the goal of our project. Yeah, it's really a lot of factor play here. It's not like I just want to. It's not just by randomly select a library. Of course, if you have enough resource and you want to screen much larger to increase the success of your project, of course, you can always expand the size of the library that you want to screen on. Yeah.
Okay, just one thing to complement what Louis is saying. Much of those selections come with experience. Like, because we know so much about this target, we can select the library that make this work. But in practice, you need to try, ask people around, try a few libraries till you find one where you have hit. Because one outcome that happens most of the time when you do this, if you screen your library, there's no hit. There's nothing above 6, nothing.
Exactly. So,
what do you do then? Change library by doing what? Learning more about your problem, going, talk to a chemist to understand, okay, what's not going well, and then doing that. There might be, in that particular case, where there's no hit, auto distribution shift, you come back to your ML modeling, and then you figure out a better strategy for model selection. There's a whole literature of papers and methods doing just that, figuring out how to best select your model when there's a big shift of in the molecular space. I hope that helped. Good.
I just want to add on that. Actually, what we see, remember for the training set, we have a maximum of ICP of 10. But here, the maximum ICPT we predict is only about 8, less than 8. So what if your project goal is to discover a much more potent molecule? Actually, this library completely failed. Also, maybe our model also failed. So again, then you have to make a choice. Okay, maybe I have to expand my library size and screen even more molecule, or completely move away from this chemical space to another. So it's really a lot of factor plays here. Yeah.
Thank you for the lab. So if you go at the beginning, you had a data frame where you showed 5 rows of the data.
5 rows of the data.
At the very beginning. Yes, over here. So I'm trying to understand exactly what we're seeing here. So we have a series of compounds that are basically inhibiting that particular protein, EGFR, and they have these IC50 values, right? And we're trying to predict the IC50 for compounds that specifically target this protein, right?
Yes.
So we've already screened some, we've already seen some drugs only for this protein, right?
Yes.
Like if we want to generalize to another protein, this approach would not work, at least with this kind of data.
Yeah, that's correct. Well, I would not say 100%, but most of the time it wouldn't work. But because here EGFR is a kinase, and in the family of kinase family, there are a lot of proteins. Maybe sometimes you're lucky, maybe somehow you can discover, but it's definitely not guaranteed.
But I'm trying to even understand what's the usefulness of having such a model, because you already have a database with so many compounds that are targeted against this gene, right? So then why would you train a model just for this? Like, why would you train an EGFR model, ML model, basically, EGFR specific?
Okay, so first of
Why
all,
would you do that?
okay, there are different scenarios. First of all, so for EGFR, there are many mutants. Like maybe previously have discovered one drug, but there are new mutant, and people start realizing, the existing drug doesn't work on it anymore. That's why you need to find new ones. Or the drug patient has developed a resistance against this drug, so we have to find the new ones which works better and last longer. Another factor here is that as you said, we have found already some very potent molecules, but not necessarily this very potent molecule really is now in like a real drug. It's already past the FDA, because those drugs might have a lot of issue like toxicity or other DMPK issues which stop this molecule move on the later stage of drug development phase. That's why we need to learn from the seen SAR, so the knowledge from these molecules, and then try to find another molecule which might success for the other factor like toxicity, ADMET property, or so just some basic thing like a solubility issue. So that's the one of the cases.
I just wanted to add one thing to that. I think one thing that I was surprised by when I started out is even when there is an approved drug for a particular disease or indication, there can be value in pursuing additional drugs that improve on quality of care. If you have a solution that requires you to be in a hospital bed 24/7 with an IV attached to your arm, that's not a very desirable situation for anyone. If you can instead target the same protein, the same indication, but with a pill you take once a day, that's a very different situation. So even if there's a drug approved for a particular disease, it doesn't mean that the problem is solved. So that's just a final sort of axis in which you may want to, you may find yourself in a scenario where there is actually data available that you can use to start this process.
Second follow-up question, sorry, super short. Why
Why target IC50 and not something like binding affinity? Is this normal for drug screening? Also
Yes,
IC50, because IC50 is like the half time. So
and that's one of the very common readout that people use to represent the inhibition ability of a drug. So why not directly binding affinity? For example, of course, if you have enough data, you can definitely use the KIKD. But in reality, this kind of oh, sorry, where am I? This kind of readout is rather up there.
So which one is better for
Definitely KIKD are better because they're more accurate, but they're also more, like the inhibition constant, for example, but they're more expensive. And also depending on the type of assay, sometimes there are cases that you don't know the target, you don't know how the mechanism work. So sometimes people use phenotype bioassay for the phenotype changes that which can also which we can also calculate the IC50 rather easily based on the phenotype changes.
Yeah.
So yeah, and this is just really much easier and accessible to use in practice.
Just
yeah.
to complement that, I think there's no right or wrong, by the way. It comes down to the resources that you have. It comes down to many other things, like even if you use KD, KI, whatever, in many projects, they are still insufficient, like, because those are not functional assays. KD tell you how strong is your binding or how strong is the dissociation, whatever. But that's not functional. Functional is, if I put the drug, what's the impact on the phenotype that I'm trying to change? Is that drug, because I put it there, the mutation that was happening before doesn't have the impact that it was having to lead to the disease? And that are a lot of other experience or experiment you need to do to get to more meaningful readout. But we need to start with something that's cheap enough and allowed to cover a large part of the chemical space. And that's why, like, sometimes we use things like IC50. It's still not cheap though. IC50 are not cheap because, as you can see there, we need many different concentrations. And the more concentration we have there, the more accurate they are. But nobody is doing IC50 with 20 concentration point. Right? So it really depends. And drug discovery, that's what Barrett was trying to convey this morning. Drug discovery is not as easy as computer vision, where we will give you straight answers, like, and you will understand. It comes with a lot of experience. And when you start working on a program, most of the time as a beginner, you will have more questions to the people you are working with than bringing them answers, for sure. And you need to do that for 5 years, then you might start giving them some answers.
Could you talk a little bit about why, so you train a model on predicting binding affinity, but ultimately with like virtual screening is a different task where you have a set of molecules where you don't know if it binds or not. Could you talk a little bit about like why train a model for binding affinity for the task of virtual screening? When the ultimate question is in virtual screening is whether or not a molecule can bind.
I didn't hear the question clearly. Did you?
Yeah, sorry. So you train a model on whether or not it can predict binding affinity, and all of your training data is you have a molecule and binding affinity. But in your screening problem, you have a set of molecules where you're trying to figure out whether or not a molecule can bind or not. Can you talk to a little bit of why use this model? Why train a model on binding affinity for this particular task when you're ultimately probably going to get a lot of false positives, as was mentioned earlier. A lot of these compounds won't actually bind experimentally.
Okay, I think if I understood correctly, so basically, because here modeling on PIC50 is just a way of how people measure the inhibition of a drug. But this, so we try to, the model is just to try to have an approximate of that, and at least we hope that the model is able to predict well, in the best scenario, predict well that value. And why we use this model? Why we use this model for virtual screening? Just want to go down. Like.
So you can, if you get us, like you can train like a classifier, for example, of binders versus not.
I mean like that.
Yeah, I mean, we also talked a lot earlier today about the appropriateness of a data set.
Okay.
So I'm just curious.
No, now I understood your question. Of course, you can always. I just use regress as an example, but in the real, there's no like a absolute solution which will always work for everything, because sometimes, depending on your data, depending on your goal, again, sometimes the regressor works better in this scenario for this data. Sometimes the classifier works better for that data. It's just so in real programs, sometimes we train both regressor and classifier and see which model can help us distinguish the active compound and inactive compound the best. Sometimes we even combine them, and then do a majority vote based on that. So it's just a complementary.
Yeah, maybe, I think if I understand your question correctly, you were also asking, it seems like we're trying to filter on something else and we're training the model on. Is that? Yeah, so I don't actually think that's the case. So like PIC50 is a measure that approximates binding affinity, and for all sorts of practical reasons, that may be the data that you have early on, right? Because it's cheaper to gather. So like we don't always have the luxury of having a data set that corresponds exactly to what we want. So there is I think we are actually, the model that we trained is aligned with the objective that we have. Yeah, I think maybe to summarize, I think PIC50 is actually a proxy or like a valuable measure at an early stage of a drug discovery program.
Maybe the point I should have made was the distribution of like
Yes, so that's very much true. And I think that's not an easy problem to answer. When we're talking about like distribution shifts, that happens all the time when you're modeling within drug discovery, that often people think about like changes in the chemical space, but you'll also get changes in the target space. So like any information you have about your downstream problem will help you come up with a way to evaluate your model that sort of like best represents that downstream setting. But sometimes you don't know, or sometimes you want to actually train on a balanced distribution because your model will train better, even if you then evaluate on something that's not entirely representative of your downstream application. So here again, it's definitely a relevant problem. There's not a straightforward solution, unfortunately, on like how you could split your data to match them. Cool.
Yeah, so in this lab, we were looking at binding affinity, and we mentioned how there is, you can predict KD or KI, or you can predict PIC50. And there's also, property prediction for solubility and so on that you can do with machine learning. I was just wondering, I don't know if there's like a rigorous answer, but are the different metrics of binding affinity equally difficult to predict for a neural network, or different in difficulty? Maybe some properties or some binding affinity metrics are actually easier for some reason, in the sense that the true binding affinity metric correlates well typically with the predicted, whereas maybe some other metrics tend to have a poor correlation because for some reason those metrics are actually harder to predict correctly. I was just wondering, if there's some sort of study even for, KD versus IC50, that's the question.
That's a good question. I think again it comes back to what we discussed earlier, like for example, certain metrics like the KD, it's just the kinetic binding, but you don't know this will really translate to the phenotype. Then maybe this is just a bad choice. If you don't know your target that well, you don't know the mechanism that well, then it's gonna fail in the end. But then if you sometimes IC50, is there cases that IC50 failed? But really depending again on the program. I think for IC50, if you are using a functional assay, again, it's really depending on if your assay is able to capture the signal you want to, which are related to your program for example, for inhibition or for activation purpose, then maybe even what you measured might not really translate to the problem itself, then it will also fail. Is there anything you want to add, Prudential?
Yeah, I mean, there's no, that's a very tough question. Like, if the target and the readout have an impact on modeling, the easy answer is yes, but so do the representation that you choose, right? If you had ultimately what have an impact on the model you learn is the input that goes into it and how it influences the optimization landscape. So there's many things at play here to define whether a particular readout is easier to learn than another. But ultimately, what Lou is saying is correct in the sense that it's not a question we ask ourselves most of the time, because like this is kind of very machine learning view. What's the easiest way I can learn that problem? We rather take the other view. What's the most impactful readout for me to kind of push this program forward, find drug that will serve the patient at the end. And once we found that by working with biologists and chemists and the budget that we have, then we figure out the machine learning part. Does that make sense? Yeah.
I have a two-part question. During one of the talks, they talked kind of about the metadata related to the experimental readout, whether it's temperature, pH, et cetera. I'm wondering, one, what is the kind of range you can expect or accept on this metadata for regression-based data and then, or, regression-based tasks? And the other side of the coin is like, let's say you're doing a binary classification and you have data across a variety of experimental readouts. Is it acceptable to use things that are, say, phenotype-based or, IC50, et cetera? I'm just wondering, what your takes are on these cases where you have not so great data to work with.
You want to take this one?
That's a good question. I think typically speaking, data creation is like super hard. I think typically speaking, as soon as you start mixing data from sources that are not generated in the exact same way, you'll start introducing noise. And the more different things are, the more noisy things are. And it's a bit of a balance, right? Like on one hand, you would like to have more data to learn from, on the other hand, making sure the problem stays relevant. There's a really nice recent paper from Greg Landrum. He did work on, he's one of the maintainers of, or the main developer of RDKit. And he did a nice paper on like just trying different curation strategies and figuring out what the effect is on the data sets and how much noise you get. I think that's a good resource to sort of learn more about the problem. I don't think there's a straightforward answer in terms of in what scenarios you can combine, you can't combine. It really depends.
So I think I just want to add, in short, always know your data and always know your bioassay. Ask people if they're relevant before you start machine learning. There are a whole bunch of homework you should do beforehand. So just be aware.
Thank you for the lab. And I would like to have one more question regarding IC50 and actually activity binary classification, as one of the people was talking about. In front of me, lead PCBA benchmark, which is constructed from PubChem bioassays. HTS screens 400,000 compounds in average per target, about 15 targets. And when I try to plot activity values in micromolar range, and tag what molecules are active and what are not, based on the author's decisions from the assays, it turns out that there is no clear cutoff where on the activity threshold where those molecules are active or not. They are completely mixed, I would say. So how could that happen? And what actually are we trying to predict then? If binding affinity prediction is not the right, or maybe it is the better choice, I don't know. How could that be difference?
Yeah, that's a interesting observation. So are you sure like there's no protocol on like for that, for example, on that page, there's nothing specify how those are computed? One of the reason maybe could be we are looking at IC50 or PIC50. Maybe you overlooked the unit of the concentration.
Oh yeah, it's basically they use 25 concentration levels of the S curve as you
Uh-huh.
were showing, and they took at 50% of the inhibition or at activation, whatever the target is. But let's say for inhibition, they use 50% of inhibition level.
So basically they use the same, either it's micromolar or nanomolar.
Yes,
for their
yeah, that's all correct. So I did not overlook that. But to your question, indeed, they use some kind of a different sense. They put some PubChem activity score, which I need to realize later what it is. Maybe the answer is somewhere there, but it looks like they made the decision not based on the binding affinity,
so they made a decision on the computed Campbell score, like that, or?
Yeah, I need to realize. Do you know how this PubChem activity score is called?
Yeah. And that's interesting. Maybe offline you can send me the essay and have a look. I think it's worth to talk to the author. It's just
Oh yeah,
yeah.
thank you.
Yeah.
Thank you so much.
By the way, our second speaker of the day, or third speaker of the day, Dominic, is quite experienced with PCBA, so he can also help you with that. One more question and we can end for the day. I think there was a hand here. No? Okay, cool. No more questions. Thanks. Let's thank again the Lou and
Thank you.
So this concludes our first day. Thank you all for attending till the end. Tomorrow we are starting at 8:30. Hopefully there will be more breaks and also more interesting topics. Tomorrow is about generative modeling. Yeah. And if you have any question, any feedback, I'll be here with Tony for another 30 minutes. Please come find us. Thank you so much and see you tomorrow.